So very good morning, dear students. So today what we will be doing is we'll be studying about the topic of body fluids and circulation. So we are going to discuss about how blood is being circulated in our body. So especially in human beings, you should remember the circulatory system is a closed type of circulatory system. When we are talking about closed type of circulatory system, it implies that blood is pumped by the heart into blood vessels. They are not pumped into the open spaces. So there are blood vessels which are carrying these fluids. So the main function of the heart is to pump the blood. Okay, so it is not like seat of emotions and all those, isn't it? So we have a lot of misconception that we are speaking from the bottom of my heart, or uh, so we have to buy heart. So many terms we use, but heart doesn't have any role in memory, neither in emotions. All our emotions and memories, they're all associated with our brain. The only function of heart is pumping the blood and circulating the blood. And it is going to help in purifying the impure blood. Yeah, what is pure and impure blood in terms of circulatory system is pure blood is oxygenated blood. We call it as pure blood. Impure blood is they have carbon dioxide more than oxygen. So the carbon dioxide carrying blood, we call it as impure blood, or one which is less in oxygen, we call it as deoxygenated blood. So you can remember it as oxygenated and deoxygenated blood. So pure blood is oxygenated and deoxygenated blood. So you have to remember about these two features. Okay. And apart from that, human heart is myocardic. So that implies human heart is myocardic and neuro and not neurogenic. It is myogenic heart. That means muscles are going to control the initiation of impulses and conduction of impulses. It is not brain which is controlling them. Already we have discussed about cardiac muscles. So they are involuntary in functions. They do not undergo fatigue. They are having intercalated discs. They are branched. Okay, they do have striations. So all that we have studied under muscular system. So here you should also remember those points regarding the cardiac muscles. Apart from that, the heart is myogenic. In human beings, the heart is myogenic. It is a muscular heart. It is controlled by the muscles. Okay, only in king crab or lemurs, the heart is being controlled by the nervous system. So we call it as neurogenic heart. Okay, the main function of the circulatory system is to see to it that the oxygenated blood is supplied to all parts of the body, and deoxygenated blood is collected from all parts of the body and brought to the heart. From the heart, the deoxygenated blood is taken to the lungs, where they get purified. So these are the uh, things that are happening in case of this body fluids and circulation. Here we are going to study the vertical section of heart, then origin and conduction of impulses. So all those and disorders of the circulatory system. So we are going to study about that in detail. So let us proceed with the commencement of this chapter, body fluids and circulation. In the backdrop, you can see that there is a electrocardiogram, ECG. Okay, so electrocardiogram, so using which we can understand uh, the functioning of heart. So there is how is the impulses initiated, conducted, everything we'll be studying under this topic of body fluids and circulation. So let us understand what is circulation. So circulation is the transport of nutrients. See, we have the from the digestion process, we get the simplest type of food, glucose, amino acids, fatty acids. They are absorbed into the bloodstream. Now, this nutrients has to be supplied to each and every part of the body, and that is being done by the blood. So from the tip of the toe to the tip of the brain, they require oxygen, carbon dioxide, and the excretory products has to be removed from this tissues also. So all that is being done by blood. So circulation is is the transport of not only nutrients but oxygen, carbon dioxide, and excretory products to the concerned tissues or organs. See, the nitrogenous waste, so they are carried to the kidneys to be purified. So whenever we are talking about purification, pure blood and impure blood in terms of kidneys, we are talking about blood having the nitrogenous waste, we call it as impure, and blood without nitrogenous waste or urea, we call it as pure. So we should understand in context, in whether it is, if in terms of heart we are talking about it as oxygenated and deoxygenated blood whenever we imply pure and impure blood. In context of kidneys, we are talking about urea enriched blood or without urea. So whenever we are talking about impure and pure blood. So let us understand about this aspect of it. The circulation is the transport of nutrients, oxygen, carbon dioxide, and excretory products to the concerned tissues or organs. So for circulation, in case of simple organisms like sponges, coelentrates, okay, phylum Porifera, phylum coelentrata, they use water from their surroundings. They don't have blood. Okay, they use water from their surroundings for circulation. The complex organisms like human beings, we have body fluids like blood and lymph for circulation. So the complex organisms like human beings, we have body fluids, that is blood and lymph for circulation. The types of circulatory system are, circulatory system is of two types: the open type of circulatory system and closed type of circulatory system. So if the blood is directly pumped into the open spaces or cavities, which are called as sinuses, so we call that as open type of circulatory system. Example, arthropods and mollusks, you find this type of open type of circulatory system. So the heart directly pumps the blood into the open spaces or cavities, which are called as sinuses. So the blood is pumped by the heart, passes through large vessels into open spaces or cavities called as sinuses. So example is arthropods and mollusks, they exhibit this open type of circulatory system. In case of closed circulatory system, here blood is pumped by the heart. It is circulated through blood vessels. So blood is pumped by the heart. It is circulated through the blood vessels. So it is more advantageous in the flow of fluid, and it can be precisely regulated. Example, annelids and chordates, they have this closed type of circulatory system. See, the annelidans like earthworm, they also have closed type of circulatory system. So the heart, here blood is pumped by the heart, is circulated through the blood vessels. It is more advantageous in the flow of fluid, and it can be precisely regulated. Example, annelids and chordates, they have this closed type of circulatory system. So this you can notice it in case of earthworm. So the closed type of circulatory system, blood is pumped by the heart, so which is circulated through blood vessels. It is more advantageous as the flow of fluid can be precisely regulated. Okay, so in case of earthworm, they also have red blood, but hemoglobin is in plasma, whereas we have hemoglobin in the uh, RBC, red blood cells, isn't it? But they have hemoglobin in the plasma. That is the difference between human blood and that of a earthworm blood, even though both are red in color. So the plasma of the earthworm consists of this hemoglobin, whereas in human beings, RBC, red blood cells, will be having the hemoglobin. So annelids and chordates, they exhibit this closed type of circulatory system. So blood is pumped by the heart and it is circulated through blood vessels. So they are more advantageous as they can precisely regulate the flow of these fluids, that is blood and lymph. So the heart of vertebrates, if you look into fishes, they have two chambered. Amphibians, they have three chambered, two atria and a ventricle. Reptiles, except cockroach, they're all, sorry, crocodiles, except crocodiles, they're all three chambered. In crocodiles, they are four chambered. So the Heart, in case of reptiles, it is incomplete partition for ventricle. So that is noticed. So except for crocodiles where they have four chambered heart, so the reptiles have three chambered heart with partially incomplete partition for ventricle. So complete partition is not there for ventricle. So they have three chambered heart with incomplete partition for ventricle. So they have two atria and one ventricle with an incomplete partition for ventricle. So the crocodiles, birds, and mammals, we have a four-chambered heart. Okay, two atria and two ventricles, or two auricles and two ventricles. So the fishes have two-chambered heart. They have a single atrium and a single ventricle. So they have a single atrium and single ventricle. So the amphibians, they have three-chambered heart. They have two auricles and a single ventricle. Okay, the reptiles, except crocodiles, even they are three chambered. So in the reptile, majority of the reptile heart, they have an incomplete partition in the ventricles. So they have incomplete partition for the ventricles. So the crocodiles, birds, and mammals, we all have four chambered heart with two auricles and two ventricles. So all vertebrates have a muscular chambered heart, myogenic heart. So heart of vertebrates is a myogenic heart. So the circulatory pathway in human beings, it is double circulation. Okay, so mammals, they exhibit this double circulation. So the patterns of circulation is single circulation. So you can notice that. Then incomplete double circulation. Okay, then double circulation. So the patterns of circulation in vertebrates is single circulation, incomplete double circulation, then double circulation, as in case of mammals. So the patterns of circulation in vertebrates are in single circulation, it is seen in fishes. So you notice that the heart receives, so they have two chambered heart, you know that they have an atria and a ventricle. So it is seen in fishes, the heart receives the impure blood, only venous heart. So they are going to receive the impure blood. Uh, so the deoxygenated blood, so so we call the single circulation as venous heart. Heart receives only impure blood. So the deoxygenated blood, they reach the heart. So from heart, this deoxygenated blood is carried to the gills through capillaries, and the oxygenated blood. So at the gills, the gill circulation, you can notice that the blood is oxygenated, and that oxygenated blood is supplied to all the body parts, and deoxygenated blood is again brought back to the uh, heart. Okay, so that is how the single circulation takes place in case of uh, fishes. See, the blood, the oxygenated blood does not again come back to the heart and it is pumped from the heart. Rather, after oxygenation from the gills through the aorta, they are supplied to all parts of the body. So they do not go back to the heart. That is the reason it is a single circulation, and heart receives only impure blood. So that is the reason the heart, in case of fishes, which is undergoing single circulation, we also call it as venous heart because it is receiving only impure blood. So they have systemic circulation and gill circulation. Patterns of circulation in vertebrates. The second one is incomplete double circulation. So this we can notice in case of amphibians and reptiles where there is no separation of ventricles. Okay, they have a three-chambered heart with two auricles and one ventricle. So it is an incomplete double circulation. It is noticed in case of amphibians and reptiles. So the left atrium, the left side of the heart. So the left atrium, they receive the oxygenated blood from gills, lungs, or skin. Okay, the right atrium gets deoxygenated blood. So the right atrium is going to get the deoxygenated from blood from all the other body parts. However, whatever the type of blood, they get mixed up in the single ventricle. So since they have a single ventricle, this deoxygenated blood and oxygenated blood, they get mixed up. So ventricle pumps out a mixed blood. That is why they have given it as the purple colored one. So blue usually represents the impure blood, where that is deoxygenated blood, and red implies the oxygenated blood. So oxygenated blood circulation is very short duration, so they undergo systemic circulation and pulmonary circulation. But in the ventricles, both the oxygenated and deoxygenated blood they get mixed. Okay, so this mixed blood is pumped by the single ventricle to all parts of the body. So ventricle pumps out this mixed blood. So that is why we call this as incomplete double circulation. We notice it in case of amphibians and reptiles. So crocodiles do not show that, but amphibians and reptiles generally they exhibit this type of incomplete double circulation, majorly because they have two auricles or atria and a single ventricle. Because they have a single ventricle, even though whether the blood might be purified, so the deoxygenated and oxygenated blood gets mixed up in the ventricle, and this mixed blood is supplied to uh, supplied by the single ventricle to all parts of the body. So ventricle pumps out this mixed blood. Double circulation is noticed in case of birds, mammals, and crocodile. Okay, the right atrium, they receive this deoxygenated blood from all parts of the body. They are going to receive this deoxygenated blood, and this deoxygenated blood, which is blue in color, it is pumped into the right ventricle. So right ventricle pumps it into the pulmonary artery. And they are going to reach the lungs where they get the deoxygenated blood gets purified. It passes on to the left ventricle. So the purified blood, left atrium receives the oxygenated blood. So this purified blood, it passes on to the left ventricle. So through the left auricle, the pulmonary veins brings this purified blood to the left auricle, and they are pumped into the left ventricle. Left ventricle is more muscular than right ventricle. Reason is it has to pump the blood throughout the body. So the aorta carries this oxygenated blood throughout the body. So you find two types of circulation here: systemic circulation and pulmonary circulation. So ventricles pump this oxygenated blood without any mixing up because they have a partition which separates the flow of oxygenated and deoxygenated blood. So there is no possibility of mix up of blood in the ventricle. So ventricles pumps it out separately, the oxygenated blood, without any mixing up. So this type of circulation, we notice it in case of birds and mammals, including man. Man also has this type of double circulation. So wherein you can notice pulmonary circulation and systemic circulation. So remember about this aspect of it. Then the next that we are going to discuss about is the human circulatory system. So as I have told you, heart is the seat of pumping of blood. Okay. Next time you put an arrow into it and other things, please remember you are causing septa or a atrial defect you are causing. So whenever there is a hole in the heart, it leads to lot of diseases. So understand that it is not a seat of memory. Heart is not a seat of memory, neither it is a seat of emotions. So next time you are speaking, I'm speaking from my bottom of the heart. Remember, you are speaking from the ventricle of the heart. Okay, so all your emotions, memory, everything is associated with the brain. It is not concerned with heart. Heart has only one role, as I told you. It is involved in pumping of blood. So it is involved in pumping the oxygenated blood to all parts of the body, and it is involved in pumping the deoxygenated blood to the lungs so that they get oxygenated. So this is the only role of the uh, heart. So remember about this. So the human circulatory system, we have the blood vessels and heart. So we are going to discuss about that. So the heart, it has the pulmonary arteries, left pulmonary artery, right pulmonary artery, aorta, which supplies the blood to all parts of the body. So the circulatory system, we'll be studying about the blood vessels and heart. So let us understand the human circulatory system. So there is two circulatory system that we have. One is blood vascular system, another is lymphatic system. So the lymphatic system, they have lymph. Lymph is the colorless fluid. They have the lymph vessels and lymph nodes. So the lymphatic system consists of the fluid, lymph, lymph vessels, and lymph nodes. The blood vascular system consists of heart, blood, and blood vessels. So the blood, it is majorly composed of the fluid part of the blood, we call it as plasma, and the cellular components or formed elements. So this blood has this formed elements or cellular components like red blood cells or erythrocytes, white blood cells or leukocytes, platelets, blood platelets or thrombocytes. Okay, so the WBC is of two types, with granules, without granules. So the granulocytes and agranulocytes. Agranulocytes have lymphocytes and monocyte. Monocyte is the largest WBC cell. So in percentage of distribution, neutrophils are larger in distribution. So the under A granulocytes, two types are there, lymphocytes and monocytes. Lymphocytes are of two types, B lymphocytes and T lymphocytes. B lymphocytes is from bone marrow, T lymphocytes is from thymus. So these are the two types of lymphocytes. Okay, so the granulocytes, based on their staining capabilities, they have the neutrophils, eosinophils, and basophils. Okay, the blood vessels, they consist of arteries, veins, and capillaries. So arteries, all the arteries carry the oxygenated blood except for pulmonary artery. So the pulmonary artery, which arises from the heart, so the which arises from the right ventricle, the pulmonary artery, it carries deoxygenated blood. All the veins carry deoxygenated blood except for one vein which carries pure blood or oxygenated blood, that is pulmonary vein. So the pulmonary vein is going to carry the pure blood or purified blood, the oxygenated blood from lungs into the right auricle. So the right auricle, not the right auricle, it is the left auricle. So they are going to bring the oxygenated blood. So the pulmonary vein is an exception. Except for pulmonary vein, all the veins carry the uh, deoxygenated blood. Except for the pulmonary artery, all the arteries carry the oxygenated blood. So then the capillaries. So they are very much smaller than the arteries and veins, and they branch to form these smaller ones. We call it as the capillaries, smaller blood vessels. Okay, so these are all the components of an human circulatory system. Human circulatory system is of two types, the blood vascular system and lymphatic system. The blood vascular system consists of heart, blood, and blood vessels. So the blood, it is having two components, the fluid part of the blood, which we call it as plasma, and the formed parts or the cellular components. The formed elements or the cellular component of blood, they have this red blood cell or erythrocyte, white blood cell or leukocytes, platelets or the blood platelets or thrombocytes. So today you know about the importance of blood platelet count, especially for the chikungunya, dengue. So they take up this blood platelet count. Okay, so the WBC is of two major types, those cells with granules and cells without granule. So granulocytes are of three types. They are neutrophils, eosinophils, and basophils. Agranulocytes are of two types, lymphocytes and monocytes. Lymphocytes are of two types, B lymphocytes and T lymphocytes. So B lymphocytes are originating from bone marrow. T lymphocytes are originating from the thymus gland. So the blood vessels are of three types, arteries, veins, and capillaries. Okay, the lymphatic system consists of lymph vessels, and lymph nodes. Blood vascular system, blood, the composition of blood is plasma and formed elements. So the plasma is 55% of the composition of blood is plasma, 45% are the formed elements. So the plasma is straw colored and slightly alkaline. So their pH is 7.4. So slightly alkaline it is, and slightly alkaline fluid matrix. So the fluid component of the blood plasma is straw colored and slightly alkaline, whose pH is 7.4, uh, and it's a slightly alkaline fluid matrix. Okay, the formed elements, they are the 45% of the blood is composed of this formed elements, that is RBC, WBC, and blood platelets. They include the cellular component. So the plasma within the body, the blood having this fluid part, we call it as plasma. Outside the body, if the blood clots, so the fluid part which is left out, we call it as serum. So after clotting, the fluid part that is left out is called as serum. But within the body, so the fluid part of the blood, we call it as plasma. Okay, let us remember about the difference between plasma and serum. So 55% of the composition of blood is made up of plasma. So they are straw colored and slightly alkaline. Their pH is 7.4. So slightly alkaline fluid matrix they are. Okay, whereas the formed elements, they are the totally 45% of the blood is composed of this formed elements like RBC, WBC, and platelets. So blood, the constituents of plasma. What is the plasma made up of? So 55% of blood is plasma, you know that. So plasma consists of water, 90 to 92% of water. Okay, we call it as plasma is majorly composed of water. So plasma without clotting factors. So plasma, this is the clotted blood, and the fluid that is collected, plasma without clotting factors, it is called as serum. Okay, so the major constituent of plasma is water, so 90 to 92%. So the function of water is it's a good solvent. So the plasma proteins, they are composed of 6 to 8%. What are the plasma proteins? So they consist of fibrinogen for blood clotting, coagulation, or clotting. We require this fibrinogen. Globulins, they act as antibodies for defense of the body. Albumins, they help in osmotic balance and they regulate the blood pressure. So the plasma proteins, they consist of fibrinogens, globulins, and albumins. Fibrinogens for blood coagulation or clotting. Okay, the globulins, they act as antibodies. So the globulins act as antibodies for defense of the body. And albumins, they help in osmotic balance and they regulate the blood pressure. So the glucose, amino acids, and lipid cholesterol are also found in the plasma. They are required for the energy production and growth. So the inorganic components like sodium ion, magnesium ion, chlorine ion, bicarbonate, calcium, regulation of osmosis. So they are all present in the plasma. So various gases are also present in the plasma like carbon dioxide, oxygen, nitrogen, etc. They are for transportation part of it. So they are for transport. So the plasma without clotting factor, we call it as serum. So here the blood has clotted and the fluid above that we are calling it as serum. Okay, so the blood vascular, blood is a river of life. I told you the important fluid which is considered to be the river of life. It helps in the maintenance of the various activities of our body. Okay, so the blood vascular system, the blood, the constituents of plasma we discussed, water is nearly 90 to 92%. The plasma proteins are 6 to 8%. They are composed of three types: fibrinogen, globulins, and albumin. Fibrinogen, they help in coagulation of blood or blood clotting. So globulins, they act as antibodies for defense of the body. Albumins, they help in osmotic balance and they regulate the blood pressure. So the plasma also contains glucose, amino acids, lipids, cholesterol for energy production and growth. You know that the plasma carries, the blood carries the nutrients. So the nutrients will be here in the plasma. The inorganic ions like sodium ion, magnesium ion, chlorine ion, bicarbonate, calcium. So they are all carried by the plasma. They help in the regulation of osmosis. They also have diffuse gases like carbon dioxide, oxygen, nitrogen, etc. for transportation. The formed elements of blood are the red blood cells, RBC or erythrocytes, the white blood cells, WBC or leukocytes, the platelets or thrombocytes. So if you look into this, the white blood cells, they are colorless. They do not have hemoglobin, so they are colorless. Red blood cells are red in color because of the presence of the respiratory pigment, hemoglobin. Okay, hemoglobin brings about the red color in case of red blood cells. They have these platelets. See, red blood cells in case of human beings, they do not have nucleus. They are enucleate. So RBC of mammals are enucleate except for one mammal, that is the camel, which is the ship of the desert. So except for camel, llama, so all the others have this uh, enucleated RBC. Camel and llama, they are going to have nucleated RBC. Okay, even the platelets, they are pinched fragments of megakaryocytes of bone marrow cells. So they are the pinched fragments of megakaryocytes of bone marrow cells. Even they do not have nucleus. Only the WBC will be having the nucleus, the white blood cells, or we also call it as leukocytes. Red blood cells or red blood corpuscles or erythrocytes, white blood cells, WBC or white blood corpuscles or leukocytes, blood platelets, or we also call it as thrombocytes. Let us understand the features of RBC, that is red blood cell. So their color is red due to the presence of hemoglobin. Normal HB level, that is hemoglobin level in blood, is 12 to 16 gram per 100 ml of blood. 12 to 16 gram per 100 ml of blood. That is the normal hemoglobin level in blood. So you know that it is a quaternary uh, protein structure. They have four hemes, so two alpha and two beta. Okay, so the lifespan of RBC is 120 days. So the RBCs are arranged one above the other like a pile of coins. We call it as Rolex. Okay, we call that as Rolex. So the lifespan of RBC is 120 days. Their count is 5 to 5.5 million per per cubic millimeter of blood. They have 5 to 5.5 million. Million is 10 lakh. 1 million equals 10 lakh. So 5 to 5.5 million RBCs per cubic millimeter of blood. So per millimeter cube of blood. So they are formed. Where are these RBCs formed? They are formed in the red bone marrow. So their structure is they are biconcave in shape. They don't have nucleus. Mitochondria, no nucleus. Mitochondria, Golgi complex, etc. Their major function of RBC, red blood cell, is to carry carbon dioxide and oxygen transport. So their major function of blood is to carry carbon dioxide and oxygen for transport and nutrients also. Especially here we are talking about RBC. So their major function is carbon dioxide and oxygen transport. The worn out RBCs or dead RBCs are destroyed in spleen. So the worn out RBCs or dead RBCs are destroyed in spleen. So spleen is considered to be the graveyard of RBCs. The worn out RBCs are uh, destroyed in spleen. So spleen is considered to be the graveyard of RBCs. So let us remember about that. So the worn out RBCs are destroyed in spleen and they are considered to be the graveyard of RBCs. So the features of WBC, so blood, so the features of WBC is what we are going to study about the next one. So the color is they are colorless. The lifespan of WBC is 1 to 15 days. So they have a very less lifespan. 6,000 to 8,000 cubic millimeter of blood. So per cubic millimeter of blood, the total number of WBC will be very less, that is 6,000 to 8,000. If their number is more WBC, then they are analyzed to be having leukemia or blood cancer. So their count, the WBC count is 6,000 to 8,000 cubic millimeter of blood. Per cubic millimeter of blood, they have 6,000 to 8,000 WBC per cubic millimeter of blood. So it is very less in number. So whereas this WBC is formed, they are formed in the bone marrow, lymph glands, and spleen. So WBCs are formed in the bone marrow, lymph glands, and spleen. So the structure of WBC is they are nucleated and they are of different types. So the functions is they are a part of immune system. WBC is a part of immune system. So based on the presence of granules or absence of granules, they are of two types. So granulocytes, which consists of neutrophils. Neutrophils is nearly 60 to 65% of total WBC is made up of neutrophils. They are the largest percentage in distribution. So then eosinophils and basophils. This is based on their staining ability. The granulocytes are classified into two types, three types: neutrophil, eosinophils, they are acidic stains, they would respond. Neutrophils, they don't respond to any stain. Basophils, they respond to alkaline stain. Okay, the agranulocytes consists of um, monocytes and lymphocytes. Monocytes are the largest WBC cell. In terms of size, monocytes are the largest. So then we also find the agranulocytes like lymphocytes. Granulocytes, neutrophils. So many times they have asked it for questions concerned with that. Neutrophils, it is also called as heterophils. So 60 to 65% of the total WBC is made up of neutrophils. So the function of neutrophils are they are phagocytosis, soldier of the body. Cell eating, we call it as phagocytosis. That is any microorganism which come across, they are going to uh, eat them up. That is, they are going to form food vacuole of them and digest them off. So neutrophils or heterophils are considered to be the soldiers of the body. Eosinophils or acidophils, they are 2 to 3%. They resist infections and they cause allergic reactions. So they resist infections. Eosinophils, they resist infections, they cause allergic reactions. Basophils or cyanophils are 0.5 to 1%. So they secrete histamine, serotonin. So basophils secrete histamine, serotonin, heparin, and they cause inflammatory reactions. So basophils are also called as cyanophils. They are 0.5 to 1%. So the basophils secrete histamine, serotonin, heparin, and they cause inflammatory reactions. So HSH, histamine, serotonin, heparin are secreted by basophils and they cause inflammatory reaction. So this is the role of basophils or cyanophils. And you should also remember the percentage. So monocytes, they are the largest WBC. 6 to 8% of the total WBC is made up of monocytes. They are phagocytosis in nature. That is, they are cell eating. Again, they are also soldiers of the body. The lymphocytes, they are the smallest WBC. So largest nucleus and smallest WBC. So lymphocytes are the smallest WBC, but they have the largest nucleus. So you can look here itself. They are the smallest WBC, but they have the largest nucleus when compared to all of the other types of WBC cells. Okay, so lymphocytes are of two types: B lymphocytes and T lymphocytes. Nearly 20 to 25% of total WBC is, see, after neutrophils, lymphocytes are the second largest uh, component of WBC. So they have nearly 20 to 25% of total WBC is composed of lymphocytes. They are of two types, B lymphocytes and T lymphocytes. The function of lymphocytes is immune response of the body, and they secrete antibodies. So they are responsible for the immunity of the body, and they secrete antibodies. So let us remember about this functions, their percentage and distribution of WBCs. So A granulocytes, monocytes, and lymphocytes. Granulocytes, neutrophils, eosinophils, basophils. Remember the percentage. So neutrophils are the first in percentage. Second is the lymphocytes. Third in percentage distribution is monocytes. Fourth is eosinophils. Last, very least distribution is basophils. So that is how you should remember the sequence of distribution based on the distribution. If you come across the descending order, so it would be the first largest in distribution is neutrophils, second is lymphocytes, third is the monocyte, fourth is eosinophils, last is basophils. Next is blood platelets we'll be discussing about. It is also called as thrombocytes. They are colorless. Blood platelets are also colorless. Their lifespan is only 7 days. So they count as 1.5 to 3.5 lakhs per cubic millimeter of blood. So 1.5 to 3.5 lakh uh, per cubic millimeter of blood. If the blood platelet counts drops down, it is going to be lethal. If it is less than 1.5, it is going to be lethal. So the blood platelets count, they are 1.5 to 3.5 lakh per cubic millimeter of blood. So they are formed in megakaryocytes in bone marrow. So where are the blood platelets formed? They are formed in megakaryocytes of bone marrow. They are non-nucleated cell fragments. So the blood platelets are non-nucleated cell fragments. They are involved in blood clotting. So clotting of blood, they are involved in. So the blood platelets or thrombocytes, they are colorless. Their lifespan is 7 days. They are 1.5 to 3.5 lakhs per cubic millimeter of blood. So they are pinched off fragments from megakaryocytes of bone marrow. Okay, they are non-nucleated cell fragments. They help in clotting of blood. So this is inactivated platelets and this is the activated platelets. They help in clotting of blood. So that is what is the function of thrombocytes. So blood vascular system, the blood coagulation, it is blood coagulation is nothing but clotting. So it is a mechanism for hemostasis. What is hemostasis is prevention of blood loss through injuries. So clotting mechanism helps in preventing excess loss of blood. It is a mechanism for blood coagulation, or clotting is a mechanism for hemostasis, that is prevention of blood loss through injuries. So that is what is the purpose of the blood coagulation. It is to prevent, it is a mechanism for hemostasis. Hemostasis is prevention of blood loss through injuries. So prevention of blood loss through injuries, it is called as hemostasis. So blood coagulation is a mechanism for hemostasis, prevention of blood loss through injuries. How is the blood clotting taking place? The steps involved are, say at the site of injury, there is a damage of the skin, bleeding takes place. So the clumped platelets and tissues, they are going to release the clumped platelets. So injury or damage, the vessel contracts, so the platelet plug, so the fibrin clot, that is how it is happening. So the site of injury, the clumped platelets and tissues, they are going to release thromboplastin and they are going to form thrombokinase or prothrombinase enzyme. And this thrombokinase enzyme in presence of calcium ions hydrolyzes prothrombin to thrombin. So they are going to activate thrombokinase enzymes are going to activate prothrombin to thrombin. And now this thrombin is involved in converting the soluble fibrinogen into insoluble fibrin. And the dead and damaged formed elements are trapped by this insoluble fibrin and they are going to form the clot. Okay, so if prothrombin is converted into thrombin, thrombin is required for converting the soluble fibrinogen into insoluble fibrin. So this insoluble fibrin is going to trap the dead and damaged formed elements, and that leads to the clot or coagulum. Okay, this is how the blood clotting takes place. So at the site of injury, the clumped platelets and tissues, they release thromboplastin. The clumped platelets, blood platelets and tissues, they release thromboplastin. So thromboplastin forms thrombokinase, that is prothrombinase enzyme. So thrombinase enzyme in presence of calcium ion, they are going to hydrolyze the inactive prothrombin into active thrombin. Once the thrombin is activated, okay, thromboplastin, thrombokinase, uh, prothrombin to thrombin. Once thrombin is activated, they are going to convert this insoluble, soluble fibrinogen into insoluble fibrin. Insoluble fibrin is going to trap the dead and damaged formed cells to form a clot or coagulum. So this is how the blood coagulation takes place at the site of injury. So you can remember it in simple steps. Okay. So at the blood coagulation, uh, coagulation leads to hemostasis. Hemostasis is prevention of loss of blood through the injured part. We call it as hemostasis. So the red blood cells, platelets, the broken blood vessel. So the activated platelets, they are going to bind. So there is a formation of clot by the fibrin. The next thing that we should be remembering about is the blood grouping. See, we have even though all our blood are red in color, we have different blood groups. Okay, especially in case of emergencies when there has to be blood transfer, you cannot transfer any type of blood to any person. So only that specific blood group has to be transferred. So Karl Landsteiner, he is Karl Landsteiner. So he was the person who discovered the blood groups. So the blood groups were discovered by Karl Landsteiner. So it includes ABO grouping and RH grouping. So RH is RH antigen, that is whether your blood is positive or negative. So I just want you to send a message in your conversation box as to how many of you are positive blood groups. Just send a message. Very good. See, majority of you are having positive blood group. So negative blood group, please send it as negative. How many of you are having negative blood groups? Negative blood group, you just send it as negative. No one is there in this group with negative blood group. None of you in the group have negative blood group. Don't feel any shy or embarrassed. It is fact. Whether you have positive or negative blood group, it's a fact. So majority of them, nearly 95% of the population, we are all having the positive blood group. And there are many hopeless people. I am also hopeless. I mean, O positive. Okay. So how many hopeless people are here? Yeah, many hopeless people are there. Yeah. I am also hopeless. So anyway, so remember about it. So see, in this present sample of your class itself, you notice that uh, not even a single person was found here, isn't it? Nearly 95% of human population, they have positive blood group. Very few of them have negative blood group. So that is why I just asked you to uh, help me in finding out the type of blood groups. So positive blood groups are, many of them are there. Okay, it includes ABO blood group and RH grouping. So O positive, to be very precise, O negative blood group is the most efficient donor, to be the best donor that you can have. See, the O blood group is a universal donor. AB blood group is a universal recipient, receiver. Okay, universal donor. So O blood group people can donate blood to all types of blood groups, whereas the AB blood group can receive blood from all types of blood groups. So that is what you can remember. So universal donor is O blood group, universal recipient is AB blood group. Okay, in that the most efficient donor is O negative blood group people. And why I wanted to know the negative blood group people was also to see to it that you find form a group of your own and know the other blood donors, so you can just have that circle of friends who have similar blood groups, negative blood groups, so that in terms or in times of emergency you can help each other out. Okay, so that is one aspect of me just asking you all these questions. So the blood groups were discovered by Carl Landsteiner. It includes ABO blood grouping. So we have, even though all our blood are red in color, we have four types of blood group. Type A, that is A blood group. Type B or B blood group. Type AB blood group, and O blood group. So we have all these four types of blood groups. Okay, and they can be A positive or A negative, B positive or B negative, AB positive or AB negative, O positive or O negative. So these are the different possibilities of blood group and their RH factor. Based on the presence of RH factor, if RH factor is there, it is positive. If RH factor is not there, they are having negative blood groups. So that is one thing. Uh, yeah, there are rare blood groups like Bombay blood group and other things are there, but they are all rare conditions. In majority of the population, you have this type of A, B, AB, and O blood group in human beings. So this was discovered by Carl Landsteiner, and it helped in progress of medicine. Imagine surgeries without this knowledge of blood transfusion. So wrong blood group, it would lead to death by complications of a wrong blood group uh, being there. Okay, so let us proceed. So you can Google it up, Sindhu and others. I'll just share it with you. So this is not required for a study. Anyway, it's a rare blood group. Phenotypes of this group lacking H antigen on the red cell membrane and anti-H in the serum. It fails to express any A, B, or H antigen on the red cells or other tissues. So that is one definition of the Bombay blood group that is uh, seen. See, AB negative is the rarest of the eight main blood types. That is also one more point that you can remember. Okay, so only 1% of total donors they have AB negative. So if any person is having AB negative blood group, you should form a community of people so that, as I told you, in case of emergency, you can help each other in donating blood. Okay, so let us proceed with this. further discussion of the topic. So ABO grouping, on what basis we have it, is based on presence or absence of two surface antigens. Okay, so they would be having two surface antigens, chemicals that induce immune response, we call it as antigens on RBCs. So they are based on presence or absence of two surface antigens. You can see here blue colored one. They are the surface antigens. They are chemicals that induce antigens are chemicals that induce immune response. So they have these two surface antigens are present on RBCs, namely A and B. Similarly, plasma contain two antibodies produced in response to antigen, namely antibody A, antibody B. So they would be having that. Okay. Now this is interesting part. So the blood group A, they have antigens on RBC is capital A. So antibodies in plasma will be anti-B, so that there is no clumping of blood. If both were, antigen was also A and antibody was A, the blood would have clotted. So in nature, we have this phenomena wherein the antigens on RBC, when they are A, the antibodies in the plasma will be B, antibody B. So they can donate blood to A and AB. They can receive blood from donors group A and O. Okay, so that is what the blood group can be done. So blood group B, they have antigens on RBCs. Okay, so the antigens on RBC that they have is B, antibody is A. Okay, they can donate blood to B and AB, and they can receive blood from B and O. Okay, so AB, so they have this antigens on them is A and B. Antibodies will be there is it is antibodyless. AB is a antibodyless blood group. They can donate blood to AB only, and they can receive blood from donors group, that is A, B, AB, and O. That is the reason AB is considered to be a universal recipient. O, they do not have any antigens on their RBC. So there is no antigens at all, antigenless. So AB is antibodyless, O is antigenless, but they have antibodies in plasma, antibody A and antibody B. So they can donate blood to all the blood groups, that is A, B, AB, and O. You might ask me, sir, they have antibodies. Wouldn't they react with the antigen of A or antigen of B? One important point that you have to remember is, so the donor's antibodies are not important. The recipient's antibodies are important. So the donor's antibodies are not important. Okay, that is the reason. So even though they have antibodies in both blood group, they do not act on these blood, because of which, so they can donate blood to, since they are antigenless, they can donate blood to all of them. So O is universal donor. AB, and O they can donate. So they can receive blood from only O blood group. So O only they can receive blood from only O blood group. So these are the different types of blood grouping that you have in case of human beings. So their antigens and antibodies, whom they can donate the blood to and from whom they can receive the blood from. That is given in this table. So ABO grouping, you should remember, antigen A reacts with antigen antibody A, antigen B reacts with antibody B. Mixing of bloods with interactive antigens and antibodies, they cause clumping or agglutination of RBCs. If you're going to mix A blood group with B blood group blood, so there would be clumping or agglutination of RBCs. So O blood group persons are called universal donors, as they can donate blood to persons with any blood group. Since they are antigenless, they can donate blood to any blood group. AB blood group are universal recipients. Since they are antibodyless, they can receive blood from any blood group. So AB group persons are called universal recipients because they can accept blood from all the groups. So the next one is blood grouping on Rh factor. So the rhesus factor or Rh factor is an antigen found on RBC. If it is present, we call it as Rh positive. If they are absent, we call it as Rh negative. So the means absence of Rh factor. So nearly 80% of human beings are Rh positive. Okay, more than that. So the nearly 80% of humans are Rh positive. See, antibody Rh antibodies uh, are not naturally found. So Rh negative person can receive Rh positive blood only once, but it causes the development of anti-Rh antibodies in his blood. So a second transfusion of Rh positive blood causes agglutination. Therefore, Rh group should be matched before transfusion. So just understand that. So the anti-Rh antibodies are not found naturally. So Rh positive, they would be having only Rh antigen. They don't have antibody. Similarly, Rh negative, they wouldn't be having any uh, antibodies. Okay, first time they can get this Rh positive blood, they can get it at the first time even though they are Rh negative. The second time, what it happens is by that time the Rh negative blood group person they will develop Rh antibodies against Rh positive. Only one time they can get transfusion from positive blood group. Second time when they go, if they take it up, it leads to agglutination. Okay, that is what it implies. Therefore, Rh group should be matched before transfusion. So Rh group should be matched before transfusion. So you can even here notice that Rh negative person can donate blood to Rh positive as well as Rh negative. But Rh positive, only once they can donate blood to Rh negative, second time it leads to agglutination. So Rh positive can donate blood to Rh positive, but Rh negative they cannot donate. Only once they can donate. So Rh antibodies are not naturally found. So Rh negative person can receive Rh positive blood only once, but it causes the development of anti-Rh antibodies in his blood. So second transfusion of Rh positive body blood causes agglutination. So it's better to avoid Rh positive blood being transfused to Rh negative blood group. Okay, therefore Rh group should be matched before transfusion. So the blood grouping, the Rh negative blood cell, Rh positive blood cell. So it is an erythroblastosis fetalis. It is an Rh incompatibility between the Rh negative blood of a pregnant mother and Rh positive blood of the fetus. Okay, so Rh antigens do not get mixed with maternal blood in first pregnancy because placenta separates the two blood. Okay, but at the time of first delivery there is possibility of exposure of maternal blood to small amount of the Rh positive blood from the fetus. This induces the formation of Rh antibodies in maternal blood. Okay, in case of a subsequent pregnancies, Rh antibodies from mother leak into the fetal blood and they destroy the fetus. So this is within the fetus, you can notice this. So in case of a subsequent pregnancies, Rh antibodies from mother leak into the fetal blood which is having Rh positive and destroy the fetal RBCs. This is fatal to fetus or they cause severe anemia and jaundice to the baby. This condition is called as erythroblastosis fetalis. Okay, erythroblastosis fetalis can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of first child. So that has to be done immediately after the delivery of first child. So a negative blood group woman should not marry a positive blood group man. Okay, but even after that, you know that uh, many couples, because of our compatibility. So all this doesn't become a important factor for them. So they do get married even though they are aware that the woman is Rh negative and the man is Rh positive. So it would be better for an Rh negative woman if she marries an Rh negative man. Okay, so there wouldn't be complications during childbirth. This is only in case of woman being Rh negative. If a man is Rh negative and woman is Rh positive, there are no issues. Okay, so the Rh negative factor, you should remember that a woman, if she has that, the first pregnancy, there wouldn't be any complications. Okay, so the second pregnancy, the complications arises, and that is because of what I have explained now. So in the first pregnancy, so the mother is having negative blood group, the fetus, and she has got married to a positive blood group man, and they would be having a fetus baby which would be having positive blood group. Obviously, it would be positive blood group. In the first pregnancy, there is no mixing up of blood. So the placenta and other connections are there. There is no mixing up of blood. And the first pregnancy, there wouldn't be any complications. So Rh antigens do not get mixed with maternal blood in first pregnancy because the placenta separates the two blood groups. Okay. But at the time of first delivery, there is a possibility of exposure of the maternal blood to small amount of the Rh positive blood from the fetus. So they would be having that exposure to small amount of the Rh positive blood from the fetus. As a result of that, this induces the formation of Rh antibodies in mother's blood. So earlier there was no Rh antibody. Now they have this Rh antibody developed, which is yellowish in color here. So during second pregnancy, what happens is in subsequent pregnancies, the Rh antibodies from mother leaks into the fetal blood, which is Rh positive, and they have this Rh antibody, and they destroy the RBC of the fetus, and this is very fatal to the fetus as it causes anemia and jaundice to the baby. This condition is called as erythroblastosis fetalis. So this condition where there is severe anemia and jaundice to the baby, and this condition we call it as erythroblastosis fetalis. So they undergo anemia and jaundice. That is what it is, and it is fatal. So the erythroblastosis fetalis can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of the first child. So erythroblastosis fetalis can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of her first child. Okay, so that is what we call it as erythroblastosis fetalis. So the blood vascular system types of blood vessels we'll discuss in the next class. Okay, so we'll wind up with today's session. Do you have any doubts or clarification, dear students?